Structure, Part 5. In addition to endomembrane structures and organelles that we might find in a typical eukaryotic cell, we would also find what's called a cytoskeleton network. The cytoskeleton, as the name implies, cyto referring to cell, skeleton, some kind of structure that gives an entity shape or form or support, is what the cytoskeleton does inside of a eukaryotic cell. In fact, prokaryotic cells also have cytoskeletal structures, though more simple in organization. It appears that almost all cells, or many types of cells, have some form of cytoskeleton structure. The network of cytoskeleton is formed of filaments and tubules. A filament is like a piece of string. A tubule, as the word implies, is a tube, like a garden hose, so it's hollow in the center. In addition to helping the cell maintain its shape, we're going to find that many organelles are positioned or attached or located along the cytoskeleton. The cytoskeleton inside of the cell not only allows the cell to maintain form and shape and give it structure and sort of strength, it also is a causeway or a pathway or a highway on which organelles and vesicles will move. There are tiny molecular motors that provide the movement or the transport along the cytoskeleton whenever it's needed. The cytoskeleton can also be used by certain types of cells for movement of the whole cell itself within a multicellular organism. So the cytoskeleton has a fairly dynamic contribution to any kind of cell in which we study and investigate the structure and function of a cytoskeleton. There are three components in a cell that are used for cytoskeletal structure. We call them microfilaments, intermediate filaments, and microtubules. As the name implies, the microtubules are hollow in the center, like a garden hose, but the filaments, both micro and intermediate, are like pieces of string or rope. They are solid structures with no hollow center. The differences would be the size. Microfilaments are the thinnest, smallest filaments or structures, and the microtubules are the larger of the three possible categories. As we see in the illustration on this slide, you can see a complex network of filaments and tubules forming the cytoskeleton of this example cell. Microfilaments are composed of a protein called actin. Actin is illustrated in this drawing as little spheres. And you can see we actually have what are called helices, or very gently wrapped structures, two long filaments of actin molecules arranged like beads on a string, sort of wrapped around each other. That wrapping or that twisting gives the actin filament some of its strength. Actin filaments also tend to be a little bit stretchy, kind of like a rubber band. So wherever you find actin filaments, you're going to find some what we call tensile strength, as well a little bit of stretchiness or flexibility. The microtubules, on the other hand, are also formed of building blocks or units, but they look dumbbell shaped. We call them tubulin dimers, and these dumbbell tubulin dimers are arranged again in linear structures, but they're sort of wrapped around in an almost alternating or sort of slightly curved arrangement. When you look at a cross-section of a microtubule, you can see the empty hollow center. Sometimes there are additional materials inside these centers, but usually we think of a microtubule as being a hollow tube with the tubulin dimers forming the wall or the outer surface of the microtubule. It's the largest of the two. Actin's the smallest. You can see the diameter of actin is 6 nanometers, where the microtubule diameter is 25 nanometers. In between the two, as the name implies, are the intermediate filaments. Intermediate filaments are formed by long linear packages or twisted linear arrangements of what are called protofilaments. These are just a little bit larger in diameter than the actin filaments, anywhere from 8 to 10 nanometers. All kinds of these filamentous structures can be used not only for cytoskeleton, but also for motility or movement of the cell. And here's an example of how we could use microtubules bundled together in different arrangements to form motility structures we might find on certain types of cells. Some of those motility structures are called cilia, which are short and very numerous on the surface of certain types of cells, like a paramecium, which is illustrated on the lower left of this drawing or illustration, or could be flagella, illustrated on the right, where we have two individual separate flagella, and the plural of that would be flagellum. When we take a cross-section or look inside either cilia or flagella, what we're going to find are arrangements of microtubules. The arrangement or pattern of the microtubules is what we call a 9+2 array. We have 9 pair of microtubules forming a circle or a long linear arrangement inside the cilia and flagella. So there are 9 outer pair with 1 central pair. The central pair is the 2, the 9 outer pairs are the 9, and they are unfused in the center. The pairs of microtubules on the outside are actually linked together by tiny molecular motors, and that's the flexing of the little molecular motors along the microtubules in the 9 pair array that causes the flexing, or what we call the beating of the cilia and the flagellum. So they have kind of a whip-like motion or a wave-like motion, and that's what gives the cell its motility or mobility. It can move around, swim around, if you will, by the flexing of these structures. For the cytoskeleton of a prokaryotic cell, as scientists are looking more and more, they're finding different types of cytoskeletal structures. Again, made by tubulin material, actin, or intermediate filaments. The tubulin material might be used in a type of cell division, where we have to split or separate 2 individual cells. The actin is also going to be used for structure, illustrated by the corkscrew green actin filament illustrated in the drawing, and also helping separate the 2 halves of the cell in cell division. And then the intermediate filaments gives the cell some of its shape or outer surface. Until scientists actually started looking for cytoskeletal structures in prokaryotic cells, it was thought for many decades that prokaryotic cells just didn't have a cytoskeleton. But when scientists started looking, lo and behold, there they were. For the molecular motors, we have 3 different types of molecular motors: kinesin, myosin, and dynein. Kinesin and dynein especially often serve as little transport devices, carrying organelles back and forth, or vesicles back and forth inside of a cell along microtubule or actin filaments or intermediate filaments. Myosin is a molecular motor we often find in muscles, causing muscles to move, muscle cells to flex and move. So these 3 little categories of molecular motors actually do have movement. They often appear to sort of walk along the cytoskeleton. Here's a more natural looking or accurate depiction of these 3 different types of molecular motors. The motor head is illustrated in blue at the bottom of all three of these example motors: myosin, kinesin, and dynein. At the top is the cargo carrying domain. So one part of these little structures carries a piece of cargo or some kind of structure, and then we have the actual little feet at the bottom that move along the cytoskeleton to transport devices, or in the case of cilia and flagella, like the beading structures or flexible structures for motility that we might see on a cell. The little motor actually sort of flexes or crawls along the tubules inside the flagellum or cilia. Extracellular structures would include material such as plant cell wall or perhaps some kind of slime layer. For plant cells, a cell wall is composed of some unique materials that include the majority of cellulose as the building blocks for plant cell walls. Cell walls are going to provide extra support as an outer structure for those types of cells. It also prevents desiccation or drying. It provides a barrier or protection from microorganisms that could infect a plant cell. And there are special openings or connections from one plant cell to another called plasmodesmata that can be found in the cell wall of one plant cell to the next. The extracellular matrix of animal cells, such as a slime layer, can be made out of three different components. A better term for this outer covering of extracellular matrix that's found in animal cells, instead of just calling it a slime layer, we could call it a glycocalyx. A glycocalyx, G-L-Y-C-O, calyx, C-A-L-Y-X, a glycocalyx, glyco referring to sugar or glucose, calyx referring to covering. It's usually made of three different components. A protein called collagen. Collagen is a very stretchy kind of protein, along with other kinds of proteins that are more fibrous or linear for support. You'll also find glycoproteins called proteoglycans in the glycocalyx. Glycoproteins are sugars and proteins that are combined to make some unique chemical structures in the glycocalyx, again making it a little sticky. And then we have linking proteins that can be found on the surface of animal cells that allow cells to be linked or attached to one another. In addition to having some kind of sticky surface found on animal or plant cells, we also have specific kinds of junctions. Plasmodesmata are unique types of junctions between plant cells. For animal cells, we have several possibilities that can link or connect one animal cell to the next, allowing tissues to form. One type of linkage could be essentially called a tight junction. The tight junction, you can see as the name implies, attaches one cell to another very tightly or snug. We can have gap junctions. A gap junction looks like a tube or a connection that has a hollow center. We also could have desmosomes. Almost looks like the cells are knitted or cross-linked toward one another. Almost like fingers of a hand linked together. And then there are hemidesmosomes, which are a little bit of sort of like a desmosome where we have some linking and maybe a little bit more like a tight junction, sort of half and half. Let's take a closer look at each one of these types of junctions between animal cells. A tight junction is a watertight seal between cells, especially when you think of cells lining the intestine. That's where you want nice, smooth, tightly linked together cells. You don't want any leakage of material from the intestinal lumen, or the center of the intestine, to the surrounding tissues. That's what tight junctions will do. Prevent leakage. When we have certain kinds of diseases that weaken the tight junctions, then we see the symptoms associated with those diseases, such as dysentery or diarrhea, where we have fluid from the tissues surrounding the intestine gaining entry into the lumen. And we have the symptoms associated with dysentery or diarrhea. Too much fluid in stool or feces. Internal surfaces of organs and body cavities are also held together with tight junctions, again, to prevent that leakage from one compartment to the next, or from one organ to the next. Gap junctions, as we saw in the initial illustration, are causeways. They're connectors between one cell and another that allow a passageway. There's a slender tube or opening from one cell to the next. It's actually an assembly of different components, different pieces, that go from one cell to the next with the opening in the middle. Gap junctions are made of protein that allow water and other small materials to pass from one cell to another. Oftentimes, gap junctions have some kind of control mechanism, so only certain types of materials can pass from one cell to the next. But it is a fairly regulated structure, because you can see sometimes gap junctions can be closed, and when needed, they can be opened, allowing that passageway from one cell to the next. Desmosomes are those tight cross-lacing or overlaying, interchanging or interlinking structures from one cell to another. They're short proteins emerging from the surface of one cell that cross over or overlap similar types of structures or proteins emerging from the neighboring cell. The name of the proteins are called cadherins, and often they're described, desmosomes, as spot welding, where you've attached or overlapped these different cadherins from one cell to the next, and they're pretty tight, they're pretty snug. They're going to adjoin neighboring cells, but give the cell some stretchiness, because these cadherins can slide a little bit or can flex a little bit from one cell to the next. So we have a good attachment of the neighboring cells, but again, some flexibility. Heart, lung, and muscle cells are often linked together or attached to one another by desmosomes using these cadherin proteins. The plasmodesmata are unique to plant cells, and again, it allows the passage of material from one cell to the next, without the cell wall interfering with that transport. Plant cell walls are very sturdy structures, and not much can easily pass through a plant cell wall. So we have to have a special causeway or a pathway for materials to get from one cell cytoplasm to the next using the plasmodesmata.